For a long time when we thought about what makes our bodies tick, it kind of felt like this straightforward relay race. DNA hands off the baton to RNA, and RNA sprints to the finish line to build proteins, the tiny machines of life.
Right. The central dogma. Simple and elegant.
Yeah, but what if that race had, a team of miniature coaches on the sidelines, whispering instructions, speeding some runners up, and maybe gently slowing others down?
Now you're talking about the complexity beneath the surface.
Exactly. We're talking about a hidden world of tiny RNA molecules, specifically these fascinating little players called siRNA and miRNA. Think of them less like the main construction crew and more like the incredibly precise foremen and quality control inspectors, fine-tuning protein production.
That's a fantastic way to put it. If our genes are the grand blueprint, then siRNA and miRNA are the master adjusters, making sure everything is built to the exact specifications, maybe even catching a faulty blueprint before too many errors are made. They're like the cell's internal editors, constantly refining the output.
Exactly. And today we're diving deep into this microscopic management team, siRNA and miRNA. We'll unpack what they look like, trace their origins, figure out their actual jobs, and explore how these minuscule molecules are already causing some, major ripples in the pond of medicine. Our goal is for you to walk away with a clear understanding of these vital components, those satisfying aha moments included, without feeling like you just swam through a, dense biology textbook.
And what's immediately striking is that while both siRNA and miRNA are small regulatory RNAs, they operate with, fundamentally different styles. Imagine siRNA as a highly targeted sniper.
Right. It's like a guided missile for a single message. And structurally, these siRNAs are usually around, 21 to 23 nucleotides long, these tiny building blocks of RNA.
Yeah. It's typically a short, double-stranded molecule with an almost perfect bullseye lock on its target, its mission, to find one specific RNA message and basically dismantle it, preventing a particular protein from being made.
They're double-stranded, clinging to each other with incredibly precise base pairing picture. 2 strands of Velcro hooking together almost perfectly. And they have these little extra bits hanging off the ends, these 3-foot overhangs.
Those 3-foot overhangs are actually quite significant. Think of them as little handles or docking bays that the cellular machinery can grab onto to put the siRNA to work. Now, let's contrast that with miRNA. While they also start as longer molecules, they end up as shorter single-stranded molecules within a cellular complex called RISC, the RNA-induced silencing complex.
Right, RISC. We'll get back to that.
And the crucial difference is that miRNAs don't need that perfect lock and key. They're more like a widespread influencer.
So if siRNA is the sniper, miRNA is more like the general raising or lowering the overall temperature in a room. It can bind to multiple different target RNAs, not with that exact fit, but more of a close enough scenario.
Exactly.
Often focusing on a small but critical region called the seed sequence at their 5-foot end.
That seed sequence is key.
So if siRNA is the sniper, miRNA is more like the general raising or lowering the overall temperature in a room. It can bind to multiple different target RNAs, not with that exact fit, but more of a close enough scenario.
Exactly.
Often focusing on a small but critical region called the seed sequence at their 5-foot end.
And because one miRNA can have these near matches with many different messenger RNAs, the instructions for making proteins a single miRNA can actually coordinate the activity of entire groups of genes. It's a much broader, more subtle level of control compared to siRNA's direct one-to-one silencing.
So siRNA is like deleting a specific line of code, whereas miRNA is like gently tweaking the settings of a whole software program?
That's a great analogy, yeah.
Okay, so we have these 2 types of tiny RNA, one a precise eliminator, the other a more general regulator. But where do they even originate? Let's delve into their backstories, their biogenesis, as the scientists call it, essentially, how they're born and processed within the cell.
Right. The story of siRNA often begins with longer stretches of double-stranded RNA. Now, that might sound a bit odd because typically the RNA in our cells is single-stranded.
Right. That's what I learned.
But these double-stranded precursors can arise in a few interesting ways. Sometimes it's from an external source, like the genetic material of a virus trying to hijack our cells, or it could be synthetic RNA that scientists introduce into cells in the lab for research purposes.
So an invading double helix of RNA sets off an alarm, like cellular defense.
In a way, yes. But our own cells can also generate these double-stranded RNAs internally. Think about jumping genes, those genetic elements called transposons. Their activity can sometimes lead to the creation of double-stranded RNA.
Ah, transposons. Got it.
Or you might have different parts of our DNA being read in opposite directions, creating RNA strands that then pair up. Even single strands of RNA that fold back on themselves to form hairpin shapes can be the starting point for siRNA production sometimes.
Okay, so we have this longer double-stranded RNA, no matter its origin.
Okay.
What's the next chapter in its life? How does it become the short sion?
Enter dicer. Dicer is this incredibly important enzyme, a real molecular tailor in the cell.
Dicer.
It belongs to a family of enzymes called RNase 3 enzymes, and its job is to precisely chop that long double-stranded RNA into those signature short siRNA duplexes we talked about, complete with those essential 3-month overhangs, those little handles we mentioned.
So dicer's like the master cutter, taking this long bolt of double-stranded fabric and snipping it into these perfectly sized little silencing tools.
Yeah.
And then what happens to these snippets?
These siRNA duplexes are then recruited by the RASC complex. We mentioned RASC before.
Right, the silencing complex.
Yeah. One of the 2 strands in the duplex, known as the guide strand, is loaded into RASC, often with the help of a specific protein called OGO2. The other strand, the passenger strand, is usually discarded. It's like the extra thread that's no longer needed.
Okay, so one strand guides RSC.
Exactly. Now, RASC, armed with its siRNA guide, goes on patrol within the cell, specifically searching for any messenger RNA that perfectly mirrors the sequence of that guide strand. And when it finds a match,
It's game over for that mRNA.
pretty much, OGO2, the catalytic heart of RISC, steps in and precisely slices the mRNA, preventing it from being used as a template to build a protein. It's a very direct and efficient way to silence a specific gene, a true one-hit wonder at the RNA level.
Wow, very efficient. Okay.
Absolutely. Now, the birth story of miRNA is a bit more intricate. It begins way back in the nucleus, the cell's control center,
Okay, starting in the nucleus this time.
with the transcription of what we call primary miRNAs or miRNAs. These are long RNA molecules transcribed by RNA polymerase to just to quickly recap, that's the main enzyme our cells use to copy DNA into RNA.
So the cell has specific instructions in its DNA for creating these miRNA precursors. It's not just random.
Exactly, not random at all. And these miRNAs have a distinctive hairpin-like structure. Imagine a bobby pin.
Okay, a hairpin.
They then undergo processing in the nucleus by a protein complex known as the microprocessor complex, which includes an enzyme called DROSHA and a protein called DGCR8.
DROSHA and DGCR8, the microprocessor.
Yeah.
Got it.
This complex acts like a molecular scissor, snipping the miRNA to release a shorter hairpin structure called the precursor miRNA, or pre-miRNA, which is around 70 nucleotides long.
Okay, so it goes from a long primary transcript to a shorter, still hairpin-shaped pre-miRNA, all within the nucleus. What's its journey from there? Does it stay put?
Nope. The pre-miRNA needs to make its way out of the nucleus and into the cytoplasm, where the protein-making machinery resides. This export is facilitated by a protein called exportin 5.
Exportin 5 sounds appropriate.
Think of it as a special VIP pass and a dedicated carrier service just for pre-miRNAs to get them across the nuclear border.
A molecular ferry. I like it. So once it's in the cytoplasm, does our friend dicer reappear? You mentioned dicer before.
It does indeed.
Dice?
Now in the cytoplasm, takes that pre-miRNA hairpin and chops off the loop part, that little curve at the top of the bobby pin, resulting in a mature miRNA duplex, typically around 22 nucleotides long.
Ah, okay.
So both siRNA and miRNA get the dicer treatment at some stage in their development, a key similarity in their processing.
That's a nice connection point, but then their paths diverge again, right, after dicer.
They do. For miRNA, one strand of the duplex is loaded into RISC, often with a slightly different Argonaut protein, maybe a GO1, compared to siRNA's usual partner, a GO2. The other strand is usually discarded.
Okay, so different AGA-GO proteins sometimes.
Potentially. Now, this miRNA-loaded RISC goes searching for its targets, those messenger RNAs with the imperfect matches, often in their 3-foot untranslated regions. Think of it as RISC having a general idea of what it's looking for, but not a precise address.
And as we discussed earlier, the main consequence of this imperfect match isn't usually slicing and dicing like with siRNA.
That's correct.
Yeah.
The imperfect binding of the miRNA to the mRNA often acts like a roadblock or a speed bump for the translation process, the creation of protein from the RNA template.
Right.
It's like putting a sticky note on the protein-making instructions, making them harder to read, or maybe just slowing the whole thing down. However, if the miRNA binding is more extensive, It can sometimes also trigger the degradation of the mRNA, just not usually through that same direct slicing mechanism that siRNA employs, more like destabilizing it.
So siRNA is the sharpshooter, directly taking out its target, while miRNA is more like a subtle conductor, gently influencing the volume of protein production from potentially hundreds of different genes. That's a pretty fundamental distinction in how they operate.
Precisely. siRNA offers that highly specific catalytic silencing 1-RASE complex can take down multiple mRNA molecules. It's a rapid and very targeted way to turn down the volume on a single gene.
Yeah, efficient.
miRNA, on the other hand, provides a much broader and more nuanced level of control, allowing the cell to fine-tune complex biological processes by affecting many targets simultaneously. It's like adjusting the overall ambiance of the cellular environment.
Okay, so these tiny RNA molecules are clearly crucial players in the inner workings of our cells. But this isn't just textbook biology anymore, is it? Carnede, in particular, has stepped out of the lab and into the clinic in a, well, pretty significant way.
Absolutely. CERN has made a remarkable transition from being a fascinating research tool to becoming a tangible therapeutic strategy. We now have approved siRNA-based therapies that are truly impacting patients' lives. It's a testament to how understanding these fundamental mechanisms can lead to real-world solutions.
That's incredible. Can you give us some concrete examples of how siRNA is being used to treat diseases? Like, what's out there?
Certainly. One of the early and really compelling success stories is patisiran. It's used to treat hereditary transthyretin amyloidosis.
Okay, amyloidosis. That sounds serious.
And a list of approved siRNA therapies is growing. Givosiran targets a gene involved in acute hepatic porphyria, a rare genetic disorder that causes debilitating attacks. Inclisiran is used to lower LDL cholesterol.
Wow, so they're using siRNA to directly target and shut down the gene responsible for the disease-causing protein. That's incredibly precise intervention at the RNA level.
Exactly. It is. It's a condition where a misfolded protein builds up in various organs, causing significant damage. Patisiran delivers siRNA that specifically targets the messenger RNA for that faulty protein, effectively reducing its production and slowing down or even halting the progression of the disease.
Wow.
Think of it as silencing the factory that's producing the faulty parts.
Bad cholesterol.
Right, a major risk factor for heart disease by targeting a protein involved in cholesterol regulation. And then we have therapies like lumasiran, nedosiran, and vutrisiran, all aimed at treating different rare metabolic disorders. It's a really exciting and rapidly expanding field.
That's amazing progress. And how do they actually get these siRNA drugs to the right place in the body? It's not like you can just take a pill containing RNA and expect it to work, right? Our bodies are pretty good at breaking things down.
You're absolutely right. Delivery is a critical aspect, a huge challenge. Many of these approved therapies rely on sophisticated delivery systems. One common method uses lipid nanoparticles. Imagine tiny, stealthy bubbles made of fat.
Lipid nanoparticles, LNPs, hear a lot about those.
Exactly. They can encapsulate the siRNA and slip past the body's defenses to deliver their cargo directly to the target cells, particularly in the liver, which acts like the body's main processing center.
Okay, so the liver is a key target.
It is for many current therapies. Another clever strategy involves attaching the siRNA to molecules like GalNAc, which is specifically recognized by receptors on liver cells, ensuring a more targeted delivery approach. It's like having a special delivery code that only the intended recipient, the liver cell, can read.
So they're essentially sneaking the siRNA into the very cells where it needs to do its work. That's incredibly ingenious. But I imagine there are still challenges and limitations to this approach. It can't be perfect yet.
There are indeed. One significant hurdle is delivering siRNA effectively to tissues beyond the liver. The liver is a relatively accessible target due to its role in filtering the blood. Getting siRNA to other organs and cell types, considerably more complex.
Right, harder to reach places.
Another key challenge is the inherent fragility of RNA. It can be easily broken down by enzymes called nucleases that are present throughout the body. We also need to be vigilant about potential off-target effects, where the siRNA might unintentionally silence other genes that are not the intended target, and the possibility of triggering unwanted immune responses. The body might see it as foreign.
Those are all really crucial considerations for the safety and efficacy of these therapies. So how are scientists working to overcome these obstacles? What's next?
Well, researchers are constantly innovating and developing new strategies. Chemical modifications to the siRNA molecules themselves, like adding specific chemical groups, can make them more resistant to degradation and reduce the likelihood of off-target binding.
Making them tougher and more specific.
Exactly. They're also refining nanoparticle formulations to improve their stability, enhance targeting to specific tissues, and facilitate entry into cells. And the development of smart targeting ligands molecules that specifically bind to receptors found only on the desired cell types holds great promise for even more precise delivery in the future.
It sounds like the field of siRNA therapeutics is in a constant state of advancement, learning, and adapting with each new discovery. Now, let's turn our attention back to miRNA. Given its broader regulatory role, how is that translating into potential medical applications, both as a treatment and maybe as a way to diagnose diseases?
Yeah, it's a bit of a different story, but equally exciting. Given their widespread influence on so many fundamental cellular processes development, cell identity miRNAs are implicated in a vast spectrum of diseases. Think cancer, cardiovascular disease, neurodegenerative disorders like Alzheimer's, metabolic syndromes like diabetes.
Wow, a huge range.
Which makes them both highly promising therapeutic targets and valuable potential diagnostic markers.
Okay, so on the therapy side for miRNA, how can we harness their power to treat diseases since they regulate so many things?
There are essentially two main therapeutic strategies being explored. If a particular miRNA is found to be overactive in a disease, contributing to its development or progression, we can try to inhibit its function using molecules called antagomers or anti-miRNAs.
Antagomers, okay.
These are specifically designed to bind to the overactive miRNA and effectively block it from interacting with its targets, basically shut it down.
Got it. And the other way.
Conversely, if a miRNA that normally plays a protective role is found to be suppressed or missing in a disease state, we can try to restore its levels using miRNA-mimic synthetic RNA molecules that closely resemble the natural miRNA and can boost its beneficial activity within the cell.
So miRNA replacement therapy, kind of.
Yeah, essentially, it's about either silencing the bad actors or amplifying the good ones.
That makes perfect sense, a yin and yang approach to miRNA therapeutics. What kinds of diseases are being targeted with these strategies?
Cancer is a major area of focus, as many miRNAs are known to act as either oncogenes, promoting cancer development, or tumor suppressors, inhibiting it. Researchers are actively investigating miRNA-based therapies for a wide variety of cancers.
Makes sense for cancer. What else?
Viral infections are another important area, as miRNAs can influence both the host's immune response to a virus and the virus's ability to replicate within our cells. And given their role in complex regulatory networks, miRNAs are also being explored as potential treatments for cardiovascular diseases, neurodegenerative conditions, and metabolic disorders where multiple genes are often dysregulated.
It sounds like miRNA therapeutics have the potential to tackle really complex diseases where targeting just a single gene, like with siRNA, might not be sufficient. Now, what about the diagnostic potential you mentioned, these liquid biopsies? That sounds incredibly futuristic.
Yes, this is a particularly exciting and rapidly advancing field. One of the remarkable features of miRNAs is their surprising stability in various bodily fluids like blood, urine, and saliva. They hang around.
They're stable outside of cells.
Yeah, surprisingly so. Even more fascinating is that the levels of specific miRNAs often change in very predictable and disease-specific ways. This means we could potentially analyze the pattern of miRNAs in a simple blood sample, for example, to detect diseases at a very early stage.
Like a molecular fingerprint.
Exactly. Or monitor their progression over time, or even predict how a patient might respond to a particular treatment. It's like getting a molecular fingerprint of the disease without the need for invasive procedures.
So instead of traditional, sometimes uncomfortable biopsies, we could potentially get a real-time snapshot of what's happening inside the body by analyzing these tiny RNA molecules floating around in our fluids. That's mind-blowing.
Exactly. And the potential applications are enormous early detection of cancers, even before they're detectable by conventional imaging techniques, identifying individuals at high risk for cardiovascular events, or even tracking the subtle changes occurring in neurodegenerative diseases like Alzheimer's long before significant symptoms appear.
Wow.
It's like having a non-invasive window into the cellular health of the entire body.
This is truly amazing stuff. So to bring it all together, it seems like siRNA and miRNA, despite both being small regulatory RNAs, have very distinct personalities and roles within the cell. siRNA is the precise gene silencer, the sniper, with a growing track record of clinical success.
Right, already making a difference.
While miRNA is the broader, more nuanced network regulator, the conductor, with immense potential in both diagnosing and treating a wide range of complex diseases.
That's a perfect way to summarize it. siRNA has really been the trailblazer, demonstrating that RNA-based medicine is not just a futuristic concept, but a current reality. And miRNA, with its ability to orchestrate entire cellular pathways, offers an even more versatile and potentially transformative set of tools for tackling human disease.
It really feels like we're on the verge of a new era in medicine, one that's becoming increasingly personalized, highly targeted, and deeply rooted in our understanding of the intricate world of cellular biology, based on how things actually work.
Absolutely. These tiny molecules, once largely overlooked, are now revealing the incredible complexity of gene regulation and providing us with unprecedented opportunities to understand, diagnose, and treat diseases in innovative ways.
It really makes you think, doesn't it? If we're only just beginning to unravel the mysteries of these tiny regulators, what other sophisticated medical breakthroughs might be waiting to be discovered within this hidden world of RNA?
The potential seems vast.
Perhaps it's worth, looking into some of the current clinical trials involving RNA-based therapies, or even pondering the broader implications, ethical maybe, of having such precise control over our fundamental biology. Lots to think about. Thanks for joining me for this fascinating deep dive.
My pleasure. It's truly an exciting field to watch.